Our first speaker today is Sonia Jackalou, and she's in the cancer training branch of the NCI. She did a PhD at Rutgers University, then she went to France to do a postdoctoral fellowship, and she came back to the States and started working with the famous cancer prevention person, Michael Sporn, and she's been here at NCI ever since. So today she's going to talk on transforming growth factor beta and lung tumorigenesis. Sonia.
In pocket system with the best.
There we go.
Okay. All right. Thank you, Terry. Um, okay. As Terry introduced me today, we'll be talking about transforming growth factor beta in lung tumorigenesis. And you might ask, why study these two topics? Well, I'll address the second question first. Lung cancer has become the most common cause of cancer death in both men and women in the U.S., and it has surpassed even breast cancer in women. Last year, there were, oops, I didn't do that. Okay. Last year, there were over 234,000 newly diagnosed cases of lung cancer in this country, and over 154,000 deaths due to this disease. So it's a really important disease. Now, with the advent of anti-smoking campaigns and anti-tobacco campaigns in this country, most cases of lung cancer now occur in former smokers, and the average age of onset of this disease is about 70 years of age. So it's a long-term disease, particularly if you start smoking as a teenager. In addition to this, well, the occurrence of smoking has been going down in this country. In Europe and Asia, the incidence of smoking and tobacco use has increased. So no matter what we do, lung cancer is going to be a disease with us for many years to come. Now, in spite of our efforts to combat and control this disease, the 5-year survival rate is still less than 15%. So why study TGF transforming growth factor beta, or TGF beta for short? TGF beta is what is called a multifunctional regulator of cell growth. It's been shown to be a potent inhibitor of the proliferation of most normal epithelial cells. It shows widespread tissue expression in mammals and has been shown to play a pivotal role in maintaining epithelial homeostasis. It's also been associated with various types of cancer, including lung cancer, and shows what we call context-dependent inhibition or stimulation of cell proliferation and neoplastic transformation. So it's important in normal cells as well as in tumor cells. So TGF-beta can be called upon to be an attractive candidate for new therapeutic intervention approaches. So that's why we want to know more about this growth factor. Now, to understand transforming growth factor beta, we have to go back to the beginning. And TGF beta has its roots in a polypeptide called sarcoma growth factor. Sarcoma growth factor is a polypeptide secreted by Maloney Marine Sarcoma Virus Transformed, MSV, mouse fibroblasts that can stimulate normal rat fibroblasts to form colonies in self-Dogger. And this was the transformation assay that Joe DeLarco and George Dodaro used to identify this polypeptide in the 70s. Now, two classes of TGF have been isolated from MSV transformed cells. One class was shown to be able to compete with epidermal growth factor, EGF, for receptor binding, and this class was called TGF alpha. The second class was shown not to be able to compete for EGF binding, but its colony forming activity could be enhanced by EGF, and this was called TGF beta. So TGF alpha plus TGF beta equals sarcoma growth factor. And this was shown by Anita Roberts and Michael Sporn in the early 80s. So following the identification of TGF alpha and TGF beta, the purification of TGF beta proceeded from at least 3 different tissues: human platelets, human placenta, and bovine kidney by 3 investigators, all at the NCI at that time. Now, what's involved in the purification of TGF beta from bovine kidney? Typically, 100 grams of bovine kidney from the slaughterhouse was extracted with acid ethanol, then centrifuged to remove the debris, and the liquid was allowed to precipitate overnight in the cold with a mixture of ether ethanol. The next morning, the precipitate was centrifuged and redissolved in acetic acid, and then applied to an 80-liter biogel P60 column for separation. 1-liter fractions were collected, and these were lyophilized and redissolved for further chromatography for identification. So typical yield was about 6 micrograms of TGF beta per time. Now, to give you some idea of the columns that were used for this purification, shown on the left are the Biogel P60 columns, and by the pink arrow is a 55-gallon drum. So these were big columns for a big purification system. Now, the assay that was used for TGF beta employed the growth of normal rat kidney cells, NRK cells, in self agar. So typically a mixture of media, serum, NRK cells, EGF, and sample was applied to an agar base plate. It was then allowed to grow for 1 week at 37 degrees and then stained. And the colonies that may have been resulted were counted using an Omnicom image analysis system shown at the bottom. Now, if TGF beta is present, colonies would grow and they would be counted if you allowed for a size on the right. If no TGF beta is present, then you wouldn't expect colonies to grow, as shown on the left of the slide. As shown on this slide is the final HPLC purification of a system, and shown by the arrow is a 25,000 molecular weight protein entity that was identified to be TGF beta. In that time, we didn't know there were more than one isoform, so it was called TGF beta. So, and Drs. Michael Sporn and Anita Roberts are credited with identifying and purifying TGF beta at the NCI. Unfortunately, we lost Anita Roberts in 2006. Okay, but we still remember her because she has a lecture series every year named after her. Now, following the purification of TGF beta, the sequence of mature TGF beta 1 was done. TGF beta was shown to be a existed the pre-pro entity of 391 amino acids containing a signal peptide at the amino terminal end, followed by a mid protein that's been called the latency associated peptide, or LAP for short, and the mature TGF-beta polypeptide at the C terminus of 112 amino acids. As shown in the picture at the top, as shown in the cartoon at the top, is the 112 amino acids of TGF-beta 1. And shown in black are the nine characteristic cysteine residues that really identify the family. Following the following the amino acid structure identification of TGF-beta, the crystal structure of TGF-beta was identified in the early 90s. This happens to be the crystal structure of representation of TGF-beta 2 because evidently TGF-beta 2 was much easier to crystallize than TGF-beta 1. Okay, so TGF-beta exists as the dimer of two identical monomers that's held together by an interchange disulfide bond, which forms a hydrophobic pocket in the center. Now, the TGF-beta superfamily contains not only TGF-beta, which exists as three isoforms in mammals: TGF-beta 1, TGF-beta 2, and TGF-beta 3. TGF-beta 4 and TGF-beta 5 has also been identified in chicken and in xenopus, respectively. And TGF-beta 4 and TGF-beta 5 are thought to be homologs of the mammalian TGF-beta 1. Now, in addition to TGF-beta per se, the superfamily also contains the bone morphogenic proteins and growth and differentiation factors, which there are quite many of, as well as the activins and inhibins. So this is what we call the TGF-beta superfamily. So to summarize, TGF-beta exists as a 25,000 molecular weight disulfide bonded homodimer. Three highly homologous isoforms exist in mammalian species, TGF-beta 1, 2, and 3. Principal sources are platelets, bone, and spleen, again in mammals. Most cells express TGF-beta and its receptors, and I'll go more into receptors shortly. TGF-beta is usually secreted in what we call latent inactive form, which has to be activated in order for TGF-beta to do its thing. And the superfamily of TGF-beta includes not only TGF-beta, but the activins, inhibitors, BMPs, and GDF. Now, the TGF-beta superfamily has been called on to be a central control module for many biological processes. This includes such processes as development, immune system function, reproduction, angiogenesis, aging, tissue repair and injury response, metabolic regulation, and proliferative homeostasis. So there are a lot of key uh, biological processes that are controlled by the superfamily. The major biological responses that are regulated by TGF-beta include it's able to inhibit proliferation in some instances, as well as to regulate apoptosis, differentiation, and immune cell function, depending on which cell system you're looking at. It's also able to stimulate the accumulation of exosomal matrix and to promote chemotaxis. Now, model for TGF-beta pathway has been derived over the years, and if you follow uh, at the top left, the blue represents the light blue entity represents TGF-beta ligand, and that could be anything of any molecule in the superfamily. What is thought that the ligand binds the type 2 receptor, shown in pink, which is constitutively phosphorylated. This combine and makes into a moiety. Then the type 1 TGF-beta receptor, shown in light pink, is then recruited and transphosphorylated by the type 2 receptor. This then makes a moiety that is then able to translocate through the cell membrane and hook up with another family of proteins called the SMADs. SMAD2 and SMAD3 is the common protein for binding to activin or TGF-beta. SMAD1, SMAD5, SMAD5, SMAD8 are active more with the VMPs. Okay, so once these bind, the SMAD2 and 3, I'll stick with the TGF-beta at this point, is phosphorylated. This then is able to bind the common SMAD4, which we call SMAD4. And when these regulatory SMADs combine with the SMAD4 protein, then we're able to translocate to the nucleus and influence transcription. Now, the whole thing could be circumvented by what we call inhibitory SMADs, and these include SMAD6 and SMAD7. They can turn off the whole system if they're allowed to do so, and if they're present to do so. Now, clinically, TGF-beta has also been shown to act in the clinic. TGF-beta has been shown to be able to act as a tumor suppressor, and there are at least 3 instances of this. For instance, germline mutations in certain parts of the TGF-beta pathway have been shown to cause familial predisposition to cancer. For instance, SMAD4 has been shown to be mutated in juvenile polyposis system. Likewise, TGF-beta pathway components have been shown to either be somatically mutated or deleted in some human cancers. This has been shown for the type 2 receptor for TGF-beta in human non-polyposis colorectal cancer, as well as the SMAD4 in pancreatic cancer. And lastly, reduced expression of TGF-beta signaling pathway components or overexpression of endogenous pathway inhibitors have been shown to be associated with progression of disease. And this encompasses the type 2 receptor, type 1 receptor, as well as SMAD7 and SCI. Okay, SCI is an interacting protein with TGF-beta and SMAD. Now, TGF-beta in the clinic has also been shown to be a tumor promoter. So TGF-beta has been, and chiefly TGF-beta 1 in this respect, has been shown to be elevated in many types of advanced human cancers and to be correlated with both metastasis and with poor prognosis. And this has been shown to occur in a large variety of cancers, including lung cancer. Now, shown at the right in the picture is adenocarcinoma of the prostate, which has been stained with the TGF-beta 1 antibody. And you can see the brown staining, and it's sitting really at the interface between the tumor and the microenvironment. Okay? So, TGF-beta in carcinogenesis, what's its role? Is it a hero or is it a villain? Well, it turns out it can be both. TGF-beta can be a proximal effector of the malignant phenotype, but it can also be a potent growth inhibitor and tumor suppressor, and a pro-metastatic factor. So it's kind of it can do any and all depending on a lot of context. Now, over the years, a unifying hypothesis has been put together where TGF-beta is able to switch from being a tumor suppressor to a tumor promoter during cancer progression. So when you have a normal epithelium, this tumor suppressor activity of TGF-beta dominates. But as you have changes that may occur in genetics and epigenetics, then you start to lose the responsiveness to TGF-beta, and you increase its expression and or activation, and then, oops, sorry, then the pro-oncogenic activities seem to take over and dominate over the tumor suppressor. So then you have invasive cancer and metastases. Now, I've been talking mainly in this first part about SMAD-dependent pathways, where the SMAD2, 3, and the common SMAD4 are involved. In addition, TGF-beta has SMAD-independent pathways by which it can operate. And this includes both the TAC pathway that incorporates JNK and MAP kinase, as well as the REST pathway, the Rho pathway, and the PP2A pathway. Now, because we're interested in lung cancer, we've been particularly interested in the RAS MAP kinase pathway. Why? Because the KRAS proto-oncogene has been shown to have an activational mutation in at least 25 to 50% of human lung adenocarcinomas. And mutation of even one allele of KRAS is able to increase the appearance of lung lesions. There has been shown to be ample crosstalk between SMAD-dependent pathway as well as the RAS-MEK signaling pathway. Activation of the RAS pathway has been shown to be able to modulate TGF-beta signaling through the SMAD. And lastly, in vitro studies, particularly in cell culture, have shown that TGF-beta can dominate over the mitogenic effects of RAS, but activated RAS, once it's activated, can override the antiproliferative effects of TGF-beta. So we're pretty confident that the KRAS pathway is involved here. Now, how can this tumor suppression, tumor promoter activity work? Well, shown on the left side of the slide is the normal tumor suppression pathway where TGF-beta can act through the SMAD pathway or through the other pathways. Now, how can this be turned off? Well, one way is by decreased production or activity of the type 2 TGF-beta 1 beta receptor. If this is decreased, it could result in tumor production. Remember that the switch between tumor suppression and tumor promotion. Another is the possible hyperactive activity of the RAS-MAP kinase pathway that could lead to tumor promotion. The third way is by decreased levels or activity of SMADs, since these SMAD proteins are very active in the SMAD-dependent pathway. And you could think of a fourth pathway. You might have a compromised function in any of the tumor suppressor activities of TGF-beta. Okay, so what we were kind of keying into the hyperactive RAS-MAP kinase pathway here. So our broad goal was to determine the role of TGF-beta in development and malignant transformation of lung epithelial cells. And this work was done in the epithelial carcinoma section of the Cell and Cancer Biology branch of NCI. Now, we had three objectives. First, to determine the effect of TGF-beta 1 deletion and KRAS mutation alone, and then in combination on lung tumor incidence and pathology. And this is going to be in mouse models. And the second, to determine the early events that occurred in development of lung lesion and how they progressed on to tumors. And thirdly, to identify potential signal transduction pathway changes that were occurring with advancing tumorigenesis. Now, we employed the three mouse models in our studies. First was the AJ mouse system, the C57 Black 6 TGF-beta 1 heterozygous system, the AJ Black 6 TGF-beta 1 heterozygous system, and the combination that we've called the TGF-beta het KRAS latent activatable system. So we asked two questions. Does lung tumorigenesis affect the TGF-beta signaling pathway? And the reciprocal question, does the TGF-beta signaling pathway affect lung tumorigenesis? Why did we use the AJ mouse model system? Well, this mouse model has been shown to be very susceptible to obtaining chemically induced lung tumors. And the lung tumors are develop in a time-dependent manner that is very similar to what we see in the human condition, going through hyperplasia, adenoma, and onto carcinoma. The carcinomas that appear have been shown to be histologically similar to human lung adenocarcinomas. And very important, the same molecular mutations have been shown to occur in both human and mouse lung tumors. And that includes overexpression of KRAS and loss of P53 expression. Now, our carcinogen was ethyl carbamate, and ethyl carbamate works through at least two different pathways. A detox pathway in which it functions, it's broken down by an esterase in water to form harmless ethanol, CO2, and ammonia. But there's also a bioactivation pathway which works through CYP2E1. And this involves a vinyl carbamate and vinyl carbamate epoxide intermediate, which results in possible binding to of an entity that may bind to various macromolecules like DNA. So to do this study, we produced tumors in the AJ mouse model system. And we took 2-month-old AJ mice, injected them with ethyl carbamate, and waited for a period of months, and sacrificed 20 mice per month, and looked at TGF-beta and the type 1 and type 2 TGF-beta receptors. Now shown here is an immunohistochemical staining of lung tumors from successively growing tumors at 2 months, 4 months, and 8 months, stained for antibodies for TGF-beta 1, the type 1 TGF-beta receptor, and the type 2 TGF-beta receptor. Now shown by the red arrows in the left and middle panel, you will see a rather intense staining for TGF-beta 1 ligand as well as the type 1 receptor in virtually all the tumors, okay? But if you look at the right panel on the right for the type 2 receptor, you'll see very comparatively light staining in the 2 months and virtually no staining in the older tumors. So this indicates there's decreased production of the type 2 receptor protein in these advancing lung tumors. And this is shown a little better in this slide here. On the left, We show these are ethyl carbamate induced lung tumors and stained for the type 1 receptor and type 2 receptor. While the type 1 receptor shows rather intense brown staining in not only the tumor shown with the pink arrow, but also the normal bronchiolar tissue shown by the red arrow. When you look at the staining for the type 2 receptor on the right of there, you see virtually no staining in the tumor shown by the pink arrow, and rather the same amount of staining in the normal epithelium. So we're showing decreased production of type 2 receptor protein in these tumors. Now we also looked at the uh, RNA patterns for these type 1 and type 2 receptor in 5 cell lines that were mouse cell lines that were derived from mouse lung tumor cell lines. And I'll call your attention to the LM1 and particularly the PCC4 cell line, which is a ethyl carbamide induced uh, mouse cell line. And here we see reduced expression of the type 2 receptor mRNA in these 2 cell lines compared to the other 3 cell lines. So we're able to show in mouse tissue as well as in cell lines there's decreased production expression of the type 2 receptor protein and mRNA in these tissues and cell lines. Now we also looked at expression of these proteins and the mRNAs for these proteins in benzpyrene-induced AJ mouse lung tumors. And shown on the top of the slides, again, I focus your attention on the arrows. We see a rather intense brown staining for the TGF beta 1 ligand as well as the type 1 receptor, and in comparison, reduced staining of the type 2 receptor in tumors. If you focus on the middle panels, this is in situ hybridization for the mRNAs for these proteins. And while you see rather intense hybridization for the TGF beta 1 ligand and the type 1 receptor, expression of the message for type 2 receptor is very low in comparison. Shown on the bottom of the slide are staining patterns for the proteins for these 3 proteins, and they're showing rather almost identical amount of staining for them. Okay, so we've been able to show in benzpyrene-induced lung tumors that there's also a decreased mRNA and protein in the tumors for the type 2 receptor. So our model reflects that decreased production of an expression of the type 2 receptor can lead to tumor promotion in this model system. Now, our next question was, does dilution of TGF beta 1 affect lung tumorigenesis? For this, we use a C57 Black 6 TGF beta 1 mouse. So, the TGF beta knockout mouse was uh, 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 born in the 90s, and while these mice are born, they tend to start to die within a week, and after about 21 days, they all are dead from adrenal wasting syndrome. So this knockout mouse is not very good vehicle for studying carcinogenesis. But the heterozygous mouse, which has only one allele of TGF beta 1, seems to thrive and go on to reproduce. So is a uh, good candidate for studying the effects of TGF-beta 1. So in collaboration with uh, Lolly D. Wakefield at the NCI, who was interested more in liver carcinogenesis than lung cancer. She treated these mice with a liver carcinogen and found indeed she got liver increased production of tumors in the livers of the mice. But oddly and surprisingly, she found that she got even more lung tumors in these mice. So she called me up and said, well, this might be a good system to look at, the involvement of TGF-beta in lung carcinogenesis. So C57 Black 6 are not really a good vehicle for studying lung tumor genesis. So we crossed these mice, the TGF-beta 1 heterozygous mouse and the Black 6 strain, with the AJ mice, which were wild type for TGF-beta 1, to derive the first generation, the F1 generation, AJ Black 6, which contained both TGF-beta 1 heterozygous as well as wild type genotypes. So we proceeded to treat these with carcinogen and look at the lung tumors that may develop. And shown here is a immunohistochemical staining for the TGF-beta 1 ligand. Okay, we first wanted to establish that these mice were behaving as we expected them for TGF-beta. So we see white prominent staining for TGF-beta 1 in the wild type, and that's on the upper left panel, as well as the in situ hybridization for showing the mRNA. When you look at the heterozygous mouse in the middle panel, there's decreased staining for this as well for the protein as well as the antisense. Okay, and if you combine the antibody with the antigen, the uh, you lose the immunohistochemical staining pattern as well as in situ hybridization. We also conducted northern blotting and competitive RT-PCR, kind of dating this project too. And compared to the bottom left of the screen, you'll see decreased expression of the TGF-beta 1 message in the heterozygous mice compared to the wild type. And if you do competitive RT-PCR, you also see reduced competition in the heterozygous compared to the wild type. So this model is performing the way we thought it would. So once again, we took 2-month-old mice. This time we had two genotypes, two groups, one TGF-beta 1 heterozygous and one wild type. Injected them with ethyl carbamate carcinogen and sacrificed them in monthly intervals and looked at the tumors that resulted. And we found increasing amounts of hyperplasia and adenomas in the heterozygous mice, shown in red, compared to the wild type shown in green. But I'd like to focus your attention more on the panel C carcinoma, because whereas tumors carcinoma started forming within 4 months in the heterozygous mice, it took at least 12 months longer before we saw carcinomas in these mice. Okay, so we show increased tumor incidence as well as multiplicity and decreased tumor latency in the TGF-beta 1 heterozygous mouse compared to the wild type litter mates. Again, we went back and stained for the type 2 receptor proteins in these two genotypes. And if you look at the hyperplasia and adenoma, you see similar staining of the type 2 receptor protein. But when you look at the carcinoma, you compare the heterozygous on the lower right compared to the wild type, you see decreased expression of the type 2 receptor proteins in these tumors of the heterozygous mice compared to the wild type litter mice. We looked at the relative levels of the type 2 receptor mRNA in lesions from the heterozygous mouse lungs that were treated with ethyl carbamate over a period of months. And when you looks at hyperplasia versus adenoma versus carcinomas, you see decreasing expression of the type 2 receptor mRNA with increasing lung tumorigenesis, again in the heterozygous mice. So the third question we asked, does deletion of TGF-beta 1 and mutation of KRAS together affect lung tumorigenesis? For this one, we employed what we call the TGF-beta 1 heterozygous KRAS latent activatable mouse. Now, to study the interplay of these two genes, the TGF-beta 1 and KRAS, we had to generate TGF-beta 1 KRAS latent activatable mice. So we crossed TGF-beta 1 heterozygous mice in C57 black 6 strain with the KRAS latent activatable mouse for KRAS. Okay, we're able to derive 4 different genotypes. We call either the double mutant, the single KRAS mutants, the single TGF-beta 1 mutant, and the wild type litter mate. Okay, and shown here is a typical mouse lung from these 4 genotypes. Okay, so shown in panel A and B are prominent pearly white mouse lung carcinomas on the lungs of the double mutant shown in panel A and the single KRAS mutant shown in panel B. Panel C and panel D, which contain the single TGF-beta 1 heterozygous mouse and the wild type litter mates, virtually showed no lung tumors, an occasional spontaneous tumor. Okay, so we next looked at the effect of TGF-beta 1 gene deletion and KRAS mutation on mouse survival, and compared to the double mutants shown in panel A, the KRAS single mutant shown in panel B had a longer lifetime compared to both the TGF-beta 1 heterozygous and wild type litter mate shown in panel C, shown in C and D. So we concentrated on our double mutants and single KRAS latent activatable mutants. They both showed decreased lifespans compared to either the TGF-beta 1 heterozygous mouse or the wild type litter mate. We looked at the pathology of lung lesions in these two genotypes and showed that increased hyperplasia and adenomas occur in the double mutant shown in green compared to the single TGF-beta 1 uh, compared to the double mutant shown in the red. And again, I focus your attention on the adenocarcinoma, and here it's a little more iffy, but we showed that there is it's roughly the double mutant seems to have greater amount of adenocarcinoma than the single latent activatable mouse. Again, we looked at staining for type TGF-beta 1 and the type 2 receptor, and showed decreased expression of the TGF-beta 1 ligand as well as the type 2 receptor in the double mutant. That's indicated by the pink arrows. Since the SMADs are important in the TGF-beta pathway, we looked at the type 2 receptor and the SMAD3 in lung tumorigenesis. And I bring your attention to the two top panels. Both of if you compare the SMAD3 expression in the wild type, the single KRAS latent activatable with the double mutant shown on the right, you see decreased expression of SMAD3 in the double mutant, which occurs much sooner than the single mutant. The wild type in the heterozygous TGF-beta 1 mouse shows virtually no change. So in the double mutant, we're seeing expedited reduction of the type 2 receptor and SMAD3 production compared to the single mutant. We also looked at the proteins for these SMADs and type 2 receptor, and essentially we saw reduced expression of SMAD4 and SMAD7 with expedited production of the type 2 receptor as well as SMAD3. We also looked at the KRAS and the KRAS interacting pathway protein RAP1 and we're able to show expedited production of both of these proteins as well. Because TGF-beta has been shown to be important in apoptosis, we looked at apoptosis in the single latent activatable mouse with the double mouse in these long adenocarcinomas. And if you look at the red compared to the green, shows very reduced apoptosis in the double mutant compared to the latent activatable mouse tumors. So the system that we've been modeling shows ability for the type 2 receptor to be decreased as well as the RAS protein to be activated, the SMAD4 to be decreased, and apoptosis to be compromised, which all can lead to advancing lung tumor production in these model systems. Now, we've done all this work to understand the TGF-beta pathway. Are we able to use TGF-beta for active use in clinical trials? Well, it's coming along slowly, but at least 5 different types of TGF-beta compounds, including antibodies, antisense oligonucleotides, receptor kinase inhibitors, and vaccines are being actively pursued by various pharmaceutical companies. Now, in addition to that, I've kind of focused the talk more on TGF-beta than on the TGF-beta superfamily, but there's also quite active investigations going on into the BMP compounds, inhibitors being used in clinical trials. So that is kind of perking its way along. These are not simple trials, and it's not really making these compounds, these antibodies, oligos, and kinase inhibitors are not that simple. And remember, because TGF-beta is so globally active everywhere in the system, that it's very difficult to try to target these moieties to be useful in preventing disease. So that's the next level of complexity, how to target these things to make them really specific. And these were the people who worked on this project, and we thank Tyler Jacks for the mice giving us the KRAS late activatable mouse. So I'll entertain any questions you might have. Yes. There are no basic questions. They're all intriguing. Okay. Mm-hmm. Well, I'll answer the second question first, okay? Because my brain works that way. Okay. I think for the second question, whether TGF-beta is a tumor promoter, tumor suppressor versus a tumor promoter, a lot depends on the context of the cell type, and what what phase the cell is in. Okay, is it a normal cell, a close to normal cell? Is it starting to become a metastatic cell? So that has a lot to do with what role it'll play, and it switch from being a tumor suppressor to a tumor promoter. Okay. Now your first question was these isoforms, three basic isoforms. Well, it so happens that there are certain tissues have more, and certain tumors have more TGF beta 1, beta 2, beta 3 than others. We've been concentrating on TGF beta 1 because it's been like the major player here, and you can get a lot of it. The beta 2 and beta 3 are a little more tricky. They're less involved. But in the chicken, for some strange reason, and I was working in the chicken for quite a while, the chicken seems to have more TGF beta 3 than TGF beta 2 or 1. Okay? So again, it has to do with more where it is and what its role is. I showed you a slide where it's involved in development and immune suppression. Well, the various isoforms are more specific for each one. I took a global approach that just to talk about it, but TGF beta 2 may be more important in immune function than TGF beta. Okay. Yes. Okay. Hmm. It's it's secreted in a inactive form, a pre-pro form, and the pre-pro form has to be essentially cut to reveal the active form. And there are specific enzymes and specific environmental conditions that enable its activation. So that's important there. And what was the other part of your question? I'm sorry. Well, it has to be in the extracellular. It's done extracellularly, and that enables it to become active and start binding to the receptor. Okay? Yes. Mhm. Mhm. Okay. No, you're right about this. We're trying to mimic as closely to the human condition as we can. There's really no perfect mouse model system that can go on there. But you're right. I mean, you're right in your context of what you're calling adenoma here. Okay. Yes, 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 exactly. Okay. Yes.
Leading on from the AJ mouse model just mentioned, you said that you had first sacrifice, you stacked 20 mice
Hmm.
after the injection of the E65 pathway. Is that because of low penetrance of the synogen, or all 20 mice present with aphasia and then?
Well, we were trying to get enough for statistical power analysis. You know. No, in the beginning, not when you're looking at 1 month, 2 months, they're not all having tumors. I mean, most of them don't have tumors, particularly.
What does the picture look like after 10
Then I get your attention to the slide where it showed A, B, and C, uh, hyperplasia adenoma carcinoma, and depending on the phenotype there, okay, the genotype, whether it's TGF beta 1 het versus wild type, you see the increase in the tumors versus nothing, right?
So white protrusion.
Hmm?
White protrusion.
Yeah. Okay, is that clear?
No, I don't know which A, B.
I'm sorry. Maybe we can talk briefly later.
Okay.
You know, I'll point you to the slide. Okay. Yes. Well, it's hard to say. I mean, I couldn't answer that. It's possible, but whatever amount you're injecting, and I wasn't aware that people were being injected with this yet. But it's possible, I suppose.
Okay, thank you, Sandra.
Okay, thank you.
So with Anton, he got his PhD at the University of Southern California. I'm a native Californian, so I know all about that. And then he did a postdoc at the Scripps Institute in California. He worked at the Caliper Life Science for several years, and then he came to NCATS in 2004. 2011, he became a branch chief, and now he's the scientific director. So his title, Early Therapeutic Discovery in Oncology and Beyond, Challenges and Opportunities.
Thank you, Terry. It's great to be here again. My journey within NIH actually started 4 floors up in this building. We were part of the Genome Institute before we became NCATS back in 2004. So it's been an interesting journey of actually helping create what is actually a startup organization within NIH. I was the 6th person hired back in 2004 to build initially a screening center, and then we expanded our operations to later stage preclinical development. And what we do at NCATS is we deal with this sort of nebulous space between the science or nature paper and the actual therapeutic intervention that helps patients. On the NCATS Advisory Council, there is a mother of a child with rare genetic disorder who actually said several times publicly, it's great that you guys publish science and nature papers, but I cannot feed my child a science paper to improve her health. So we need actual tangible interventions. So how do you think about that process? There is actually a very sort of naive way of thinking about it, which is to think about the disease mechanism, how to intervene, what to intervene on. It may be by inhibition of individual unique enzyme, kinase in cancer, for example, or a pathway. Then you have to create a test system to discover therapeutic candidates. You test many chemicals in that system. That is an example with small molecule drug discovery, but you can imagine how it would work with, say, therapeutic antibodies through phage display and other techniques. So you run a screen, you test multiple candidates, and then in the small molecule case, you actually have to modify them through extensive chemistry to improve their properties, prepare them for in vivo use. And then you conduct tests in animals, and eventually you test the candidate molecule in humans. So it seems very linear, it seems very sort of automated, and it seems like it's somebody else's job, right? Pharma does it, they will somehow do it forever and problems will be solved. So you look at the reality, though, expressed in the bottom two graphs. On the left side, we see the great news, the science papers, the explosion of knowledge in terms of actual molecular mechanisms associated with more and more diseases. That is, we believe we understand the basis for actually well over 6,000 diseases right now, and the number is growing, but only about 500 come with therapy. And at the pace of current drug approvals worldwide, it may take about 2,000 years to address all diseases. So that's pretty long time. And then on the right side, you look at the sort of financial aspect of this operation. The productivity based on unit of R&D investment has been going down pretty steadily, and this plot is actually called Eroom's law, so Moore's law spelled backwards, indicating that it's following the opposite trend of computer chip density with time. So how do you actually reverse these trends and improve the pace of therapeutic development? That's what we try to address at NCATS in disease-agnostic manner. We cannot afford to work on one disease at a time, and we're actually not equipped to do that. So we're focusing on these overall problems that are disease-independent, such as problems in predicting the safety and efficacy of new drug candidates. They work great in cell-based assays, in isolated protein assays, maybe in mouse model, and then they fail in humans for both efficacy and toxicity reasons. How come we couldn't predict that earlier and saved probably $500 million in unnecessary preclinical development? How can we actually scale the approaches to find treatments for more than one disease at a time? Can we probe the proteome in some scalable manner to maybe bundle diseases by molecular basis, not by symptoms or names? There are many other problems in the space, and you will notice some of them are related to clinical, others are preclinical. Some of the problems are scientific in nature, others are actually organizational in nature. How do you incentivize work in this space where the team is probably 100 individuals, and you're not going to get a first author paper. What is the currency that operates in the space? It cannot be a PI-driven research. How about this as a problem? It's organizational, it's not scientific, actually. And there are many other problems, such as IP disposition, clinical trial design, and so on. So I'm going to talk primarily about the first bullet, dealing with the preclinical early discovery, and giving you examples of how we try to tackle inefficiencies in this space and how it may apply to actual projects with specific examples from oncology and elsewhere. So the journey typically starts with these high throughput screens. You design test system to look for the lucky drug candidate out of, say, 1 million test molecules, and you can set up large robotic screening systems and run these screens. And over the past 20 years or so, high throughput screening has become pretty standard step in the drug discovery process. And up in Rockville, those of you who plan on visiting our shop will see the system in operation. It's been running for 13 years, but from day one, we actually tried to improve the underlying process of high throughput screening. And one sort of glaring deficiency of this process is typically pharmaceutical companies and elsewhere, people have been testing the compound libraries, these drug candidates, at a single dose, maybe in duplicate, maybe at N of 1, depending on their budget and assay complexity. But we know well pharmacology means the effect depends on the dose, right? There is the Hill equation, there is dose response curve associated with typical drug effects. So why not do the obvious as shown on the left? Test the library, the drug candidates in multiple doses. So we actually designed and implemented a workflow to do exactly that. We used really top-of-the-line miniaturization techniques to shrink the assay volumes to 2 to 3 to 5, maximum 8 microliters per test. Developed brand new informatics pipeline to analyze, say, 500,000 dose response curves in a time span of about 20 minutes with minimal manual intervention. You know well, GraphPad Prism will not allow you to do that. You'll have to labor pretty extensively over a dozen curves. And what do we get in the end? We actually get pharmacological profiles instead of statistical hits buried sometimes in the noise. So reliability went way up. False positives and false negatives were reduced dramatically, and we were able to actually rank molecules based on whether they're inactive, just flat response, or shallow curves indicating more unique pharmacology, high potency, low IC50, weak potency, high IC50, etc., from that robotic screen. That actually helps you save money downstream on not having to follow up on a ton of false positives. We're part of NIH. Part of our mandate is to actually make data public. We've deposited pretty much every single data point that we've generated over the years, so several 100 million data points from several hundred screens. They're all available for others to analyze, build predictive models, and eventually improve the process of predicting what chemicals will be active against new targets. So again, this is disease agnostic, and source code and all the platform details have been made available for others to replicate this process. Probes that target different pathways and enzymes are not discovered directly from screens usually. You need to improve them through chemistry. So we have in our place an integrated medicinal chemistry operation. chemists work not only to improve potency of the molecule against the target. So this is actually an example of the very first in class highly selective inhibitor of ubiquitin-specific protease 1, USP1. So chemistry essentially yielded 100-fold improvement in potency relative to initial screening hit. But also, the operation allows for multi-parameter optimization for things like solubility, metabolic stability, and other physicochemical properties that are required for successful downstream animal experiment and eventually human clinical trial. So we've developed about 200 such high-quality, highly selective small molecule probes to study novel therapeutic concepts. So basically the dark matter of the human genome. Everybody tends to study the same 5 to 10 top kinases. What about the rest? Everybody tends to study kinases, GPCR, ion channels. What about the other target classes? So that's kind of how we've positioned ourselves over the years. And as a result, we've actually opened up entire segments in terms of novel target classes and therapeutic hypotheses. And some of these probes are actually further developed as drug candidates, and I'll be giving you a couple of examples. So as one example, we were actually the first shop to run high-throughput screen to discover activators for human pyruvate kinase M2. That was right around the time when the Warburg effect, the essentially the redox regulation in cancer was coming to the fore. So it was literally 10 years ago with very little knowledge in the space. Through high-throughput screening and chemistry, several different chemotypes were discovered and shared with actually several hundred labs around the world. This is an example of an early paper with Luke Antley on initial profiling of these molecules. But look at the other example in the lower right. So this is very recent. PKM2 activation was actually associated with control of metabolism in pancreatic beta cells, indicating that these molecules may actually have utility in managing diabetes. So why am I showing this? Because you don't know where the journey will take you. PKM2 was understudied target. There were no activators for it. So this therapeutic hypothesis on the lower right was not available to guide us in terms of ultimate utilization of these activators, but prospective development of such activators allowed the biology to be studied in these new therapeutic applications to be discovered. Another example from more recent work is something that a traditional medicinal chemist would not like to see, which is discovery and development of irreversible covalent inhibitor of an enzyme. Typically, one thinks of these as being highly promiscuous and not worthy of development because they would tend to hit multiple targets in vivo. But in this case, thyrodoxine reductase was again one of these understudied potential targets in oncology involved in control of redox balance, clearing of reactive oxygen species, but there were no inhibitors for the enzyme that were selective for it. And importantly shown in the second bullet point here, when we actually ran the robotic screen and found the new inhibitor that is highlighted here, it was actually highly selective and did not hit glutathione reductase, which is very similar enzyme, again controlling the redox balance, but which is essential for all the cells in the body. So you do not want to hit glutathione reductase. So essentially the therapeutic hypothesis, the hitting thyrodoxine reductase for therapeutic benefit in oncology could not have been tested without selective inhibitor that does not hit glutathione reductase. So when this molecule was tested and profiled, it was selective, and even though it was acting by covalent mechanism, why could that be? How can it be? Normally, if something is covalent, it'll hit the other related targets. So it turns out there is unique selenocysteine residue in thyrodoxine reductase that is responsible for the catalytic mechanism, and the compound was essentially acting with that hot residue, but the compound itself was not reactive against thiol cysteine type enzymes. So on the right, you can actually see some in vivo results in compound being tested against several models in vivo. This was published last year, and actually it is treated as a drug candidate through a spin-off company out of Karolinska Institute in Sweden, with whom we collaborated on this. Something that was published just a few weeks ago. You go after single target, you develop great molecule, it goes in people, it shows good results initially, and then resistance develops. So this is the case with FLT3 kinase inhibitors for AML. The team here performed in-depth profiling of the cell's response to this inhibition and discovered that cells were basically activating innate immune pathways through IRAK1/4, and that was essentially creating a mechanism for escape from FLT3 inhibition. So to tackle this problem, you don't want to be having a situation where you treat the patient with FLT3 inhibitor, wait for resistance to develop, and then try to treat with IRAK inhibitor. Our group actually developed a dual-acting molecule through extensive medchem work to target both kinases simultaneously. And you can read the paper. The molecule is shown on the lower right. It actually performed better than the current FLT3 inhibitor in several xenograft models, essentially suppressing the activation of this escape pathway from the get-go. Of course, it remains to be seen what will happen upon prolonged treatment. That we don't know, and the molecule hasn't been tested in humans because it has to go through preclinical development, but initial results are pretty encouraging. So what do you do when you don't have a molecular target, right? This is the case of so-called phenotypic screens or assays. So here's an example. This little feature called perinucleolar compartment, barely visible in this image, was discovered. It's a nuclear type organelle or feature that appears primarily in transformed cancer cells, and its prevalence is correlated with cancer spreading with metastasis. So how do you go about discovering modulators of perinucleolar compartment to test the hypothesis that targeting it would affect metastasis when you don't know what this PNC organelle is composed of? You don't have individual targets, you don't have a pathway, so there is no obvious target. So an approach that one can take in this case is literally to screen for disassembly of this feature, for compounds that make it go away. A very sort of naive approach, but you can kind of see how this would work as a discovery path. So we assembled a team of experts. Again, we're not experts in perinucleolar compartment. We're not oncologists, but we're very good at screening, and actually including someone from NCI, you may recognize Udo Rudloff. So long story short, we ran this phenotypic screen, literally scoring cells for whether they retain perinucleolar compartment after compound treatment or they don't. And the class of compounds was actually discovered, disrupted the PNC. And further studies showed that the compounds had no cytotoxic effects. So final chemically optimized molecule, which the group called metarestin, kind of a cute name, somebody worked really hard to come up with it, actually demonstrated all sorts of benefits in different mouse models of cancer metastasis. It's not as easy as it sounds. You have to really try to distinguish between cytotoxic effects, something that just kills cancer cells and shrinks tumors, the way many therapies would do, versus something that you're after here, which is affecting metastasis specifically. So a lot of work on the model systems had to be done, and Udo Rudloff actually did a lot of the heavy lifting. But you can see, for example, on the left side, the separation between cell viability affected metarestin and its PNC prevalence, and then cancer spreading. So where is this program? It's actually still in-house. This is such a risky therapeutic hypothesis that you cannot just say, I'm going to patent metarestin and I'm going to just license it and Pfizer will just buy it overnight and work on the development. Companies see this paper, the Science Translational Medicine, as too risky of an asset, too unproven. Obviously, the team is very proud of having published Science Translational Medicine paper. Companies wouldn't touch this asset. It's still too risky. So where we're at is at NCATS, we're actually supporting the preclinical development of this molecule, coming up with better synthetic strategy to scale it up, to make large amounts of it under GMP environment, and completing the IND-enabling tox studies in several animal species, working on formulation. The goal is to apply to the FDA in a few months to begin what's known as investigational new drug process to enable phase 1 safety trial and then efficacy assessment. And the goal is to actually do the proof of concept clinical trial here with NCI support, with Udo Rudloff being the lead clinician. So kind of switching gears, I talked about discovery of single type modulators, small molecule inhibitors, etc. We know well there is value in finding drug combinations to tackle resistance, to tackle various disease scenarios. 2 or 3 drugs. There are many examples of such combos from the HIV world. How do you go about rapid discovery of such combos? If you start pipetting drug A against drug B manually or using some semi-automated equipment, the numbers will just overwhelm you. It'll end up being so expensive and so slow, you're not going to be able to test practically anything. So we worked pretty hard in this space to first develop various well-annotated libraries of selective molecules and approved drugs, and then couple those with emerging technologies, in this case, acoustic dispense, allowing you to literally spot combos of 2 or more agents pretty rapidly without using pipette tips or intermediate dilutions in the time span of a couple of hours for several thousand combinations, and in parallel develop informatics platform to process all that data, calculate indices of synergism, and display all that for further examination by by the scientists. You can imagine, again, where this is important. Resistance affects both the oncology field, cancer, and the infectious disease field. Why? Because development of resistance is a function of doubling time for a cell and mutation rate. And when you start mapping various species, cancer cells, and various infectious agents, they're all kind of mapping together. You can get resistance not just in malaria or bacterial infections, you get resistance in cancer. So solving this problem is a platform problem of how to discover synergistic combinations of two or more drugs rapidly, cheaply, can affect several fields that sometimes don't seem to have anything in common. So we worked on developing this and showing proof of concept first in oncology application in this rare blood tumor with Lou Stout's group actually several years ago. Paper is shown down on the lower left. But again, it's just a paper, right? So why am I showing it? It's actually because of the bullets above it. We made source code and all protocols for this process publicly available because the goal is not just to publish PNAS paper and go home and declare victory, but to actually enable others to practice this operation in their own labs without having to knock on our door for every little detail. On the infectious disease side, just one example of several developed over the past few years. This is work with people at the clinical center here. You probably heard the stories of multi-drug resistant Klebsiella outbreak a couple of years ago at the clinical center. They killed several patients. So pretty close to home, right? So when someone presents with an infection that appears to be multi-drug resistant, how can you tackle that in real time? So we try to develop proof of concept for this. So starting with a single agent screen, we pick top hits from the individual approved drugs and drug candidates, and then combine them in binary combos at 6 to 10 concentrations each, A versus B, and came up with several synergistic combos. So left shift means synergism, green is the combo, blue and black are the individual agents. The whole process was actually published in this paper. The group actually took this one step further and tested 3 drugs, so progressing from single agent screen to dual agent, binary combos, and triple agent combos, because you can't test all 3 against each other, and came up with several combos that were highly effective against pretty much all 10 common multi-drug resistant isolates. So are we there yet? We're not there yet, but the hope, the dream is that you can actually one day set up a screening operation that is so rapid and efficient that you can take a sample from a patient and run these tests and hopefully within less than 12 hours deliver actionable suggestion or list of drugs that can be combined to tackle these infections or solid or liquid tumors. So switching gears again, I mentioned earlier, we're not very good at predicting efficacy in humans because typically we use easy to grow HEC293 HeLa cell type models in 2-dimensional microtiter plates. And then when we get to humans, we don't know why the molecule is not working. So one way to tackle this problem is to develop more sophisticated 3-dimensional models of healthy and diseased tissues. Healthy tissues to assess toxicity, diseased tissues to evaluate efficacy. So you're familiar with the concept of spheroids. You may have heard about organs on chip. These are entire, um, um, another name for them is microphysiological systems. So essentially microfluidic chips or cartridges that contain various cells and organ systems connected to each other, recapitulating sometimes pretty complex physiological processes. Um, and you look at the plot sort of in the middle. As you move from left to right, your physiological complexity increases. So organ on a chip is pretty complex. Next thing on the right that is not shown is the animal and human, right? On the left, we have very high throughput. We can test many, many samples. I mentioned earlier, we can test millions of samples at our place within less than 1 week. So clearly there is a trade-off, right? So what we're working on right now is actually the thing in the middle, trying to essentially build a triage funnel that will connect or present levels of complexity or triage between easy to execute 2D screens and the limited throughput of organs on chip or animal or human experiments. So the thing in the middle is actually 3D biofabrication or 3D tissue bioprinting. So this is a very sort of naive cartoon showing how it's supposed to work on a very basic level. Cells are suspended in hydrogel polymer type matrix, and they're loaded in one or more syringes. And the bioprinter is essentially a 3D printer with precise XYZ control of these syringes and extrusion devices. So cells are essentially extruded, controlled by CAD software, layered in various ways. The construct is incubated for several days to mature, and eventually you get a piece of tissue. It's not as simple as it sounds. You have to optimize every single step pretty extensively, and everything is new. This is basically 2 to 3 year old technology, so a lot can go wrong. And again, cells don't just stay where you place them; they move around, etc. So again, a lot of work has to be done to actually come up with a decent protocol to make these tissues. So we've been working on several tissues, actually more than what's shown here, but these are kind of the most prominent: retina, blood vessel wall, skin. Again, collaborating with tissue experts, utilizing our technology strengths and their tissue expertise. So you can imagine you first learn how to build the wild type or healthy tissue, and then you can begin to think how to layer on top of it disease models. So skin is actually the first tissue that sort of graduated in terms of being most validated. So left shows image from the internet, from the anatomy textbooks on skin that is stained. Right side shows the 3D bioprinted skin from our group. Pretty good resemblance. We have, of course, other ways to qualify the skin, its barrier function and others. And a lot of technology development has to go into figuring out how to image the outcome, the biology in these tissues. You can't just stick the piece of tissue that is several millimeters in size under a microscope and see what's inside. So our group has been working on developing tissue clearing techniques where you treat these tissues after your experiment with clearing agents to remove fatty acids and other components and allow light to penetrate the tissue. And then if you have, say, GFP reporter, your imaging system will see that reporter deep inside. Otherwise, it would be hidden, masked by the fatty acids and other components, and then you can begin to um, do what's called segmentation and rebuild the tissue depending on what markers you've embedded, whether they're um, um, fluorescent fusion proteins or others, or whether you stain with antibody. How do you fill these bioprinters with enough cells? You can't just buy primary cells for every type of tissue. So we're actually working pretty heavily on stem cell technologies. Opened a stem cell lab about 2 years ago to work on the boring part. How do you scale up iPS cell cultures? How do you automate that? How do you make the scale up cheaper? How do you make the iPS differentiation protocols into mature lineages of cardiomyocytes or various liver cells or neuronal types, cheaper, scalable, reproducible? Every day we see more and more iPS differentiation protocols get published and no one can reproduce them. And they're definitely expensive. So the lab is actually able to follow cells at the single cell or population level, to characterize them, to develop better markers for what constitutes a mature lineage. And in parallel is working to improve the protocols to produce these cell types. And we actually have several success stories there that are being published, but I don't have time to talk about them today. All sorts of reagents we're developing that are based on cheap small molecules that are able to replace expensive protein reagents like transcription factors and media components. That's the only way to enable regenerative medicine and 3D model building using stem cells as source for the cells that will go into these models. Otherwise, the math just doesn't add up. You cannot finance such projects, both in pharma and academia. I want to switch gears one more time and talk about this sort of other problem in drug development, which is hard to predict toxicity. That rears its ugly head and contributes to about a third of the late stage drug failures. So that's a pretty big problem. How come we cannot predict if something will be toxic in humans based on cell assays? There is another angle to this problem of predicting toxicity, and it's shown in this other bullet down below. There are about 80,000 chemicals. So those are usually not drugs. There are other kinds of chemicals in commerce worldwide, and most of them have no toxicological data because there is no agency around the world that demands such toxicology evaluation. And these are molecules to which we're all exposed by touching pieces of plastic, drinking water from plastic bottles, etc., etc. Very few of them have been thoroughly characterized, and a complete, say, rat tox study is extremely expensive. So no one's going to test these chemicals and actually prepare these packages, period. And no one forces industry to do that. So also there is pressure to minimize animal testing and use of animals in say, cosmetics development, etc. So there is drug discovery angle, and there is environmental safety and prioritization angle. So wouldn't it be nice to be able to come up with a panel of cell-based, relatively cheap, high-throughput assays that represent major readouts or hallmarks of toxicity, test all these chemicals, drug candidates, environmental chemicals in them, and build predictive models for how the next chemicals will fare and whether they will be toxic in humans or not. That way you can inform the chemical industry better, and you can become better at predicting whether your drug candidate will fail or not. So it's basically the mother of all systems biology projects when you think about it. Can you deconstruct the organism into constituent pathways and then using that in vitro cell-based data rebuild the human or the animal? Pretty ambitious. We started this as a partnership with several government agencies, with EPA, NIHS down in North Carolina, part of NIH, ourselves, and later on FDA joined the partnership. And this little flow chart on top shows the project in very sort of basic, naive way. So you have to develop a collection of chemicals. What are you going to test? You have to come up with these tests, the assays, representing readouts for something bad that happens to an organism or a cell. You have to generate very high quality data, and hopefully using that data build predictive models. Initially, those models will predict assay level activity, and later on, hopefully, if you have enough pathways interrogated, maybe you'll be able to rebuild the human one day. So we built a collection of about 10,000 chemicals. These comprise both approved drugs and some failed drugs, as well as non-drug-like chemicals, pesticides, industrial chemicals, and really a wide range of molecules. It was really a unique collection because nobody had assembled drugs and non-drugs type chemicals in the same collection before. So we had to actually develop quite a few new processes for handling these molecules. And on the lower right, I'm showing an example of QC LCMS trace for a compound. Because there were so many weird molecules in this collection, it could not be QC'd using the same standard method. So we had to apply gas chromatography, NMR, and other methods. But importantly, all of that QC data are now in the public domain. You practically never see that in data sets. People will show the bioactivity, sometimes cherry-picked, whatever worked, and purity separately. In this case, we're showing the activity of the compounds and their quality as tested at time 0 and after 4 months storage for people to begin to understand the effect of storage on bioactivity. On the cell assay side, we've run over 50 screening campaigns. This collection was actually arrayed at 15 doses in triplicate. So each compound was tested, essentially producing 45 data points in each screen. And so what was screened actually? So some low-hanging fruit, estrogen receptor assay. It's well recognized the endocrine disruptors are bad. So we actually ran several different versions of the estrogen receptor assay, several other nuclear hormone receptor assays, readouts of mitochondrial toxicity, such as mitochondrial membrane potential, mobilization of DNA damage response pathways, etc. All of that data are in the public domain, and again, I don't have time to go into detail, but to date, we've deposited almost exactly 100 million data points in the public domain. So what, right? Interestingly, this middle bullet point is the so what point. People all over the world have actually used this data to build predictive models using machine learning, sharing their models, and as a result, some models are 90% accurate in predicting whether a new structure will be active or inactive in, say, the estrogen receptor assay. the EPA is moving towards using the in vitro estrogen receptor assay for regulatory purposes, and we're hoping that other assays will follow. And many other organizations like public-private partnerships in the European Union and Big Pharma are actually using the data for building their own models. So I'm going to wrap up by just showing you this slide that you may find of interest as sort of further reading. I gave you a number of examples, but again, the overall theme is we're trying to improve the processes. So the individual examples are great, but if they don't relate to your work, you probably wouldn't care. So this is actually where we put a lot of the know-how, the guidance. We call this Assay Guidance Manual. It actually started about 13 years ago as a partnership with Eli Lilly, the big pharmaceutical company. They released its internal manual for assay development, screening, and early drug discovery in the public domain. And we worked with them over the years to essentially enhance the content, make it more user-friendly, and now we're approaching 1,500 printed page equivalent to this electronic book. So it's constantly refreshed and updated to pretty robust editorial team. We have chapters on pretty much all aspects of early drug discovery, starting from assay design, sources of artifacts, data analyses, and moving towards things like pharmacokinetics, drug metabolism, etc. On the right is actually something else that we're doing, which is to have series of workshops where there is actually lectures on the various topics. We recorded most of these, and if you want to watch the videos, they're all available there. And the little red plot shows the semi-annual retrievals. So this is actually a pretty popular resource for those who are interested in working in this space, how to design an assay that is better than just using luciferase reporter. Or if you do use luciferase reporter, what should you be paying attention to, etc. What is Z prime factor? What is standard deviation, etc. And we're hoping to add content in terms of stem cell-based screening models or actually regulatory affairs, clinical considerations, etc. I will end with something forward-looking. I showed you a lot of automation in the areas of biology. I mentioned machine learning in informatics, robotics, etc. I didn't talk about chemistry. Very little has changed in the chemistry lab over the past century. Maybe people are more compliant in terms of using safety goggles and gloves, but chemical synthesis is still manual operation by and large. So we're actually beginning to work in this space, and you can read little white paper we published late last year, utilizing machine learning to predict chemical reactions and predict outcomes and design routes for chemical synthesis of new chemical matter, and to validate automation modules to actually physically conduct the chemical reactions, separation steps, purification in an automated fashion, catalog as many reactions as we can, put all of that in the public domain, and essentially demystify organic synthesis so the people can actually use their brains and not their hands so heavily in the area of MedChem. I will stop here and be happy to answer questions. Thank you. Yeah.
about the combination? Because I agree it's very important. And um, and in this case also, if there are NIH-funded clinical trials, you might have the power to affect which combinations are tested. So what I'm asking is, if the FDA will approve protocols, so the one who initiates the trials are usually the company, and often they would choose a combination that also is valuable in financially, introducing the two compounds and how we find what they want to gain the indication. So, do you have access in the space of clinical trials funded by NCI to promote combinations that you have your indication that might be fruitful but are not or compounds of competing interests?
Well, if a company initiates a trial, they're not going to ask us ahead of time what's good and what's not good. Remember, there are also considerations coming from tumor sequencing, from other types of experimental platforms that can guide you towards particular combination. One big challenge that I didn't go over in this space is growing up the tumor right to fill the plate and actually run that test. With blood tumors, it's relatively easy because the cells continue to divide. You can actually have access to enough material to run these combos, but a lot of solid tumors, you can take biopsy and what do you do next? The cells most likely are not going to divide. So it's actually a pretty challenging space where you can't just take a biopsy, snap your fingers, grow up the cells, and come up with combos. So as more and more solid tumors become demystified, as in people find ways to dissociate the cells from that solid mass and find ways to grow them and show through RNA-seq and other methods that they actually retain the original tumor's signatures and don't become something else irrelevant, we can access more and more models and run more screens. And actually, we do this with NCI investigators all the time, I would say. But it's hard to intervene in somebody else's upcoming clinical trial because of the organizational issue of non-sharing. People are going to do what they want to do for their own, purposes, right? But again, scalability issues coming from being unable to grow up every type of tumor or what's sort of slowing down the field right now.
Perhaps you could comment on new small molecules which you've developed for rare diseases.
So for rare diseases, we actually don't focus on just small molecules. We work on pretty much all therapeutic modalities, including gene therapy, because it's late stage development and actually a lot of rare diseases are single mutation based and they're prime candidate for gene therapies where you can actually hope to literally cure the disease. So we actually are dealing with an interesting case where we work with a company in the space of rare disease, particular aromatic amino acid decarboxylase deficiency, where part of the therapy is injecting the gene therapy, the nanoparticle containing the vector, into specific part of the brain of very young children. Hopefully you can catch them by age of 1 or 2. So it just goes there. It's a single injection, stays there, works there, and the results are actually pretty amazing. Normally, the natural history shows that these kids will be that by age of 5 they absolutely fail all developmental milestones. They can't walk, they can't speak, they're sort of in vegetative state. Those who get treated with the therapy are pretty much like normal kids by age of 3. It's like night and day. So the clinical trial was small scale and run in Taiwan. So think the organizational challenge, will FDA accept that data? So that's where we did the heavy lifting, preparing, kind of helping with the data analysis, preparing the package for the FDA, and they actually accepted the clinical trial from Taiwan, and we're proceeding with BLA, with biologic license application to essentially qualify a manufacturer and begin to sell the therapy, to deploy it. There is no requirement for US-based trial, which saves enormous amount of resources, and hopefully we'll get to an actual deployment soon. Yeah. One can set up types of read-through assays to find compounds that will facilitate the skipping of such stop codon. Many diseases actually originate from such problem. It just happens in different genes and different, parts of a gene, etc. But the basic idea is to find compounds that will affect the transcriptional and translational machinery to allow the skipping of that stop codon and continue the read through, and hopefully generate a gene product and eventually a protein that is at least semi-functional. It may have a mutation there or something, but in some of these diseases, even a 5% improvement in terms of level of gene product is enough to dramatically actually affect the natural history. So you don't need to correct 100% in many cases. 5 to 10% may be enough to achieve significant therapeutic benefit. Yeah.
That'll do it. Thank you.
Thank you. Right.